Biology Part 82. Successful organ transplantation from one individual to another is much more difficult to accomplish than successful blood transfusion. This is because the transplanted organs like the heart or kidney contain many more antigens than either the blood plasma or the surface of the red cells, so that they are rejected by the body's immune system. In transplant surgery, this problem has been partly overcome by careful tissue typing, which matches the donor and recipient antigens as exactly as possible by the use of immunosuppressive drugs. These techniques are most effective for kidney transplants. For reasons explained in our tutorial, the interior of the eyes shows no immune response. Consequently, lens and corneal transplants are successful even between unmatched individuals. Excretions and osmoregulation. Well, now we talk about the homeostasis. The term homeostasis is used to describe the maintenance of the body of a constant internal environment. This concept has a particular meaning for multicellular organisms because only these organisms possess the internal fluid medium which is kept separate or distinct from the external environment. This fluid, called extracellular fluid or tissue fluid, acts as the internal environment on which the individual cells of the body depend for all their requirements. Homeostasis is placed multicellular organisms at a considerable advantage because it provides their body cells with relatively constant conditions. Consequently, they can tolerate a much wider range of external conditions than single-celled organisms, which remain at the mercy of even small changes in their environment. Effective homeostasis is involved in the regulation of the temperature, osmotic concentration, and detailed composition of tissue fluid. As the surrounding of the activity of the organism changes, so continual adjustment must be made. Homeostatic mechanisms are therefore dynamic systems which act to produce the state of balance. Described as dynamic equilibrium, typically the balancing process involves three functional parts. As you can see, that receptors constantly monitor the internal environment and pass information to control center. This initiates the correct response by sending out the nervous or hormonal messages. The effect of these signals upon the responding organs or consequently upon the tissue fluid is detected once again by receptors, providing feedback control so that the correcting response ceases when optimum conditions are restored. One important homeostatic mechanism has already been described earlier, and that is the maintenance of the constant blood sugar level. The subject of this tutorial is the balance of water and dissolved solutes in the body. Two major processes are involved in maintaining this balance, namely excretion and osmoregulation. Excretion is the removal of the body's waste products, metabolism, substance surplus to the body requirement. Osmoregulation is the process which controls the relative quantity of water in dissolved solutes in the tissue fluid so that their osmotic concentration remains at the appropriate level. The mechanism and structures which carry out these processes are closely related, as well as will be seen in the following sections. Excretory substances. New substances are continually acquired by the body cells by uptake from the extracellular fluid or as the products of metabolism. Similarly, the excess quantities of some chemicals may be present in the diet. Some of the substances are useful, but others, called excretory substances, are useless. or even damaging must be removed. The main excretory substance of a mammal and their origins, destinations are listed below. Number one, of course, is carbon dioxide. Carbon dioxide is formed in all body cells as a waste product of aerobic respiration. As explained earlier, it diffuses into the tissue fluid and enters the blood, where a series of reversible reactions enable it to be transported around the body, eventually reaching the lungs. Most of the body carbon dioxide is excreted at the lung surface into the alveolar air spaces, but a small proportion follows an alternative course and is excreted by the kidneys in the form of hydrogenated carbonate ions, HCO3. Nitrogen waste from protein metabolism. Surplus amino acids in the diet cannot be stored in the body but are broken down by the liver through the process called deamination, described earlier. In this process, the nitrogen containing NH2 amino group of each amino acid molecule is removed. Ammonia is formed. This substance is highly toxic and must be removed immediately. Small organisms such as amoeba, hydra can excrete ammonia by simple diffusion across their body surface. But most larger animals must convert it into less harmful substances such as urea, which are allowed to circulate in the body before being excreted. In mammals, urea is formed in the liver by a series of reactions known as ornithine cycle. It enters the bloodstream and is finally excreted by the kidneys. A small quantity of urea is also present in sweat. Other organisms, notably the main insects, some reptiles, and all birds, excrete uric acid. This substance is solid and insoluble, so that very little water is needed to eliminate it from the body.